So in our big project on algae, in order to determine what kind of algae we had, we first isolated DNA from the algae, then we PCR'd a piece of that DNA, namely we PCR'd a piece of the 18S ribosomal RNA gene to make millions and millions and millions of copies of it. From there, we send it out for sequencing. And that means determining the exact sequence of bases in that fragment. That's what we can compare in a database to known sequences of known algae to determine what kind of algae we have. So the process of figuring out the order of the bases is called Sanger sequencing. And that's what we're going to talk about today. So just first, a little reminder from lecture. When you're building a new strand of DNA, the new nucleotide that comes in has to bind to that 3' OH group. If there's no 3' OH group there, then this base cannot be put into, you can't add anything to the new strand. You'll terminate this new strand right at that point. So what Sanger noticed was that there's a very similar molecule called dideoxy. In this case, NTP, the N stands for whatever base you want it to. Notice here's the normal 3' OH group, and in this case, that 3' OH group is gone. Without that 3' OH group, nothing will be able to bind to this base. So if this particular base got incorporated into the new strand, the strand would end with this base because nothing could be added to it. So the next observation he made was this. Let's say this top strand here isn't the unknown. You don't actually know the sequence of it, okay? First you use a labeled primer, and you do this via PCR, so this is in the artificial setting, but you make a reaction with di-D-ATP in this case. And that means that when the DNA polymerase gets to the first T, it will potentially add in a dideoxy A instead of a normal A. And so you get a strand that's this long. And sometimes the DNA polymerase will grab a normal A in that position, but then grab a dideoxy A later on. And same goes for this one and this one. So which base, whether the polymerase picks up a dideoxy or a normal D-ATP, is a random chance. So sometimes it's going to pick up a normal one, sometimes it's going to pick up a dideoxy one. That generates an entire family of fragments, all of which end in A. Next thing you do is create 4 separate reactions, one with dideoxy G and all of the normal DNTPs. In other words, you have to have some normal DGTP in there. And then one for each of the 4 bases. Each reaction tube is going to generate that family of fragments of varying lengths, such that all of the reactions in tube 1 end in G, and all the ones in tube 2 will end in A, etc. So let's take this as an example. Again, imagine that this top strand is the unknown. You don't know what it is. So you generate your 4 reactions, one with dideoxy G, A, T, and C. You generate a family of fragments that ends in G, a family that ends in A, a family that ends in T, and all of these would end in C. Now, you have generated, out of all 4 tubes, you've generated pieces of DNA that end at every position in your unknown. In order to figure out which one goes where, you simply run it on an agarose gel. The same way we did in lab, what we've talked about in lecture and in the lab video. Remember that the smallest fragments move the fastest. So in this case, in the dideoxy TTP reaction generated the very smallest fragment. So you know the very first base is T. Move up one, and you've got yourself a C. And then a G, and an A, and an A, and another G, and an A, and a C, and a G, and all the way back over to T, and then it looks like an A, and a T, and a C, and you'll notice that's the sequence of your unknown. Now, excuse me, getting over something. The way they originally did this was by using radioactively labeled primers. And so when you did it that way, you would run these out on a gel. You wouldn't be able to see the bands until you expose that gel to a piece of X-ray film and then develop the X-ray. But the process here is the same. You read it from the bottom. Here we have a GG, let's see, TT, T, A, T, A, and you read that all the way up. Since working with radioactivity is touchy, we've discovered a better way to do it. And the better way to do it is to use fluorescently labeled bases so that each base gets a different color. And then you run it through what amounts to a spectrophotometer. It's a long tube that has a gel-like substance in it, like agarose gel. And when you run them through, the smallest pieces run the fastest. So the very fastest fragments run through the spectrophotometer first, and you read A comes first. You can see that peak corresponds to A, and then this color corresponds to G, and this color corresponds to T, and then a C, and you work out the sequence the same way. This is turns out to be a much faster way to do it, and has been able to allow for high throughput sequencing.